Hi, I'm Josué Ballesteros, Application Development Scientist at Lunaphore. In the Immunology 2025 annual meeting, I had the opportunity to present a poster in the spatial resolution of Alzheimer's disease using automated high-plex immunofluorescence. I'd be happy to share more about the project. Alzheimer's disease is marked by toxic protein buildup, nerve cell loss, and memory decline. But how brain cells interact during this process is still unclear. Using spatial proteomics, I mapped key cellular interactions driving the neuroimmune response, offering new insights into Alzheimer's disease. Lunaphore's spatial biology solutions were used, including the COMET platform for multiplex image acquisition and Horizon software for image analysis. COMET enables sequential immunofluorescence by performing rapid cycles of staining, imaging, and antibody elution. Horizon provides an intuitive workflow for image visualization, cell segmentation, and spatial feature analysis. FFP tissue sections of human Alzheimer's disease and healthy brain were stained and imaged on COMET with a high-plex assay consisting of 16 key biomarkers targeting amyloid-β and brain cell types such as neurons, astrocytes, microglia, and vasculature components. Here you can see composite images showcasing a selection of protein biomarkers with SEC-IF staining on the same slide of FFP human brain tissue. Accumulation of extracellular deposits of amyloid-β protein forming amyloid plaques was observed in the cortical gray matter of the Alzheimer's disease brain, with a concomitant decrease of the neuronal marker NeuN compared to healthy brain. In these stainings, astrocytes are identified as GFAP-positive cells, microglia as IBA1 positive cells, and neurons as NeuN-positive cells. Amyloid-β plaques are present only in the Alzheimer's disease brain. In both gray and white matter of the Alzheimer's disease brain, increased expression of astrocytes as GFAP-positive cells and microglia as IBA1 positive cells was observed, suggestive of neuroinflammatory phenotype that includes astrogliosis and microgliosis. A closer look at the markers expressed by the cells interacting in proximity with the amyloid-β plaques revealed an increased abundance of astrocytes expressing GFAP and microglia cells expressing HLA-DR and CD68, markers typically associated with reactive activated microglia. Using Horizon software, in situ single-cell features were detected through nuclei segmentation and cell approximation by expanding nuclei annotation masks. Then, with a user-defined thresholding of amyloid-β, I perform a supervised classification on a region of interest in the cortical gray matter of the Alzheimer's disease brain sample. In the left panel, amyloid-β immunostaining is shown in magenta. Nuclei-segmented cells classified as amyloid-β positive classes are outlined in magenta, while amyloid-β negative classes are outlined in blue. In the right panels, amyloid-β immunostaining is shown also in magenta, and HLA-DR positive and NeuN positive cells are outlined in green, while HLA-DR negative and NeuN negative cells are outlined in red. Once cells were classified, I perform a neighborhood analysis for each amyloid-β positive or amyloid-β negative classes. The heat map visualization showed the mean distance for each segmented amyloid-β positive or amyloid-β negative class to its two nearest neighbors from each defined class. This analysis revealed that amyloid-β plaques are, on average, closer to cells identified by neuroinflammatory markers, such as HLA-DR, CD68, TSPO, and GFAP, compared to amyloid-β negative classes, while other cell types, such as NeuN-positive neurons, did not show preferential localization near the amyloid-β plaques. In conclusion, a high-plex proteomics panel was developed to characterize the spatial localization of Alzheimer's disease-associated protein aggregates, alongside with key cell populations including neurons, astrocytes, microglia, and vasculature components. This analysis identified inflammatory activation of microglia and astrocytes in the proximity of amyloid-β plaques. This spatial approach offers novel insights into the spatial relationships between neurodegenerative lesions and the neighboring cells, shedding light on the neuroimmune landscape of Alzheimer's disease. If you want a closer look at my poster, you can use the QR code and download it. Thanks a lot.